As we continue to look at the function of plasma membranes, we're now going to dive in a little bit deeper and look at how particles can move along a concentration gradient or be moved actively in and out of a cell. So let's look first again at this concept of diffusion from an area of high concentration to an area of low concentration. In some particles, the particles are small enough that their movement, their passive movement by diffusion, can simply take them through the phospholipid bilayer. So hydrophobic molecules, small uncharged particles, or like oxygen molecules, for example, can simply diffuse across this plasma membrane by moving right through the phospholipid bilayer itself. This is how, for instance, oxygen taken up by your body occurs. The air that we breathe contains oxygen. It's routed down to the little sacs, the alveoli in your lungs, where they cross a cell membrane and then cross another cell membrane to get into the capillary before they're picked up by those hemoglobin molecules in our red blood cells. Very simple, just moving by diffusion. Some particles move passively along this concentration gradient by using helper proteins. And so we call this facilitated diffusion because their movement based upon the concept of diffusion as a passive transport mechanism is facilitated or helped by these transport proteins. And there's two types: a channel protein, which we see here, which is a protein whose shape, and remember how important shape is to proteins, the protein shape literally creates a little passage for these particles to move from an area of high concentration to low concentration. While a carrier protein works sort of like a revolving door. The carrier protein allows for a particle to bind to it, and when the binding happens, the carrier protein changes its shape, and the particle is allowed to move on its concentration gradient to the other side. So both channel proteins and carrier proteins are examples of passive transport by the concept of diffusion. But what about instances where particles need to be transported backwards along a concentration gradient? Passive transport moves these particles based upon their random movements from high concentration to low concentration. But what happens if the cell needs to maintain an ultra high concentration of some ion, for instance, or electrolyte on one side of the cell's membrane? In order to move these things backwards along a concentration gradient, we're going to use a form of active transport. And active transport is going to require that the cell use cellular energy in the form of usually ATP. Now, just a quick refresher about our ATP molecule, adenosine triphosphate. This is a molecule that has 3 phosphates to it. When we remove one of those phosphates, we liberate some energy that can be used to do other things, as we'll see in the next chapter. But the removal of a phosphate from ATP creates a molecule of ADP, adenosine diphosphate. So pay attention as we see how this sodium-potassium pump works as a classic example of active transport. This sodium-potassium pump works along our neurons and helps our neurons to maintain a polarized state, or a state where their membrane is charged to allow them to generate a electrochemical impulse, and this bit of electricity is how information is passed. In order for the neurons to do this, they have to remain polarized. And the sodium ions and the potassium ions simply just want to move based upon the concept of diffusion to one side or the other. But to maintain the polarity, we have to pump the sodium ions out and the potassium ions in. So let's take a look at how this works. So here is our transport protein, and the first step is where a molecule of ATP binds to the transport protein, and we see that the transport protein can accept 3 of these sodium ions. When the sodium ions are bound to the transport protein, the phosphate is pulled away from our ATP, creating a molecule of ADP. This causes a fundamental shape change and therefore a structural functional change in this protein, and the protein opens up to the other side, and this reduces the ability of these 3 sodium ions to remain attached to our protein, and so they're pumped out. Now, this change also allows for 2 potassium ions to bind to this protein, and as the potassium ions bind, the phosphate is removed. This changes the shape of the protein again, and we see the protein open up back to the inside, and these 2 potassium ions are moved out. And once again, an ATP binding to this can start the cycle all over again. You need to make sure you understand this mechanism of the sodium-potassium pump. Learn it as a mechanism, a step-by-step process, and everything should be just fine. The key points to remember is that it requires ATP, and the reason ATP is required is because we have to phosphorylate our pump. This phosphate taken from the ADP phosphorylates this pump and activates it, right? When the phosphate is removed or dephosphorylated here, that's when the potassium ions are pumped out. So 3 sodiums are pumped out, 2 potassiums are pumped in.